Hi everyone and welcome. In this video I would like to discuss a topic that often shows up in biological problems. Let's say that we're interested in the correlation that exists between, for example, the genome size and the GC content. So on the one hand, we have a couple of measurements of the genome size, and on the other hand, we can measure the percentage of Gs and Cs in the genome. And we are interested in whether or not there is a correlation between the size of a genome and the amount of Gs and Cs in the nucleotide content. So one way that we could do this is fundamentally to do a regression analysis. So we would have one of these two variables here being the X variable and the other one being the Y variable. So we would try to make maybe a scatter plot here with the variables X and Y, and we hope that the points will be maybe along a line. And for example, in this particular case, we would say that there is a good relationship, a good linear relationship between the variable X, that could be the genome size, and the variable Y, which could be the GC content. So overall, regression is a standard strategy to know whether there is a correlation between a quantitative variable and another. So what's the problem if we work with something like the genome size and the GC content? Well, we have to work with species, with entire genomes, which means that they are the product of evolution. So let's see a little bit what would happen if we were to draw the evolutionary tree of the different species we're interested in. So it would have a certain tree structure here with an ancestor and maybe 3 data points. We would have species A, B, and C, and we would measure the GC content and the genome size of species A, species B, and species C. What we would see maybe is that the 3 points are kind of well aligned, and we would hope that this would mean that the GC content is indeed positively correlated, for instance, with the genome size. But there is one big problem with this approach, is that the data here for species A, B, and C are not independent. So you could say, well, that doesn't matter so much, right? So after all, what matters is whether yes or no, there is a linear relationship between the points. Now let's consider what happens if there is a new species that is added in this tree here. And this new species is added here at the very end of the tree. And we could say that the split is extremely recent, such as, for instance, 2010. And that is caused by human activities. So species A here has split very, very recently and created an offshoot species that we will call D here, which is split since 2010, only a couple years of evolution. And so when we add this particular point here on the graph that we have, if we say that, for instance, species A was this point here, well then we can bet that species D is going to be pretty much on top of the point for species A. And the question that we're asking is, does this give us new information, yes or no? Does the fact that we just discovered species D and the fact that this is indeed a different species from species A, does this give us new information about the problem at hand here? On the one hand, you could say yes, because we have one more data point, and one more data point is always more information. On the other hand, you could say no, because in reality, there was not enough time for A and D to diverge, and therefore whichever information we already had about species A is going to be the same about species D. In other words, we do not believe that just a couple of years is enough to have any significant change, neither in genome size nor in GC content. So in reality, we have 4 points here, but the argument would be that it counts as only 3. So we see that if the split between A and D is extremely recent, there are many reasons to believe that we have the same amount of information as if we had only one species. And as the split between species is further back in time, we have more and more information, but the information is not completely new either. So here we have a certain evolutionary time between species A and B, and that time is of course longer than between A and D, but still, genome sizes don't vary so fast, and GC contents either. And so depending on the amount of evolutionary time that you give here between species A and B, you may either have almost exactly the same genome size and GC content, or you may finally allow for some variation of one of these variables, or both. So essentially we're saying that when it comes to problems that are taken from evolutionary trees, we have less information than the total number of points available in the data. One way to think of this process is to imagine that we have on the vertical axis here time that would go from top to bottom, and on the x-axis we would have whatever we're measuring. So that could be one of the two variables here that would be anything of interest to us, like the genome size or the GC content. We would start on the ancestor here that would split into two branches, and we could follow the variable x over time on one of the two branches. So on the left side, it would kind of go randomly left and right, depending on whatever processes occur on this branch. And then here, it would split again, and we would have on the left side some variation, on the other side some other variations that goes random. And if we go back to the ancestor, then we could follow the right branch. I would see maybe something similar with some variations at random, a split at some point, and then some variations somewhere, and other variations on the other side here. And so we would end up with 4 points here for the extant species at the end of the process. But we see that depending on when the split happens, the 2 points actually have exactly the same value until that point. They are allowed to diverge only over that length here of the tree. And the same for these ones. The divergence goes only over the time that they have separated. And if we measure the difference between, let's say, that species and that species, the time of divergence is longer. It goes from the root down to the species over there. So what matters here, we see with this example, is the time that has passed since the point have split, because that allows some divergence to take place. If there is not a lot of time since this divergence, then we know that the points have to be together, and if there's a lot of time since the divergence, then the value of x can be actually very far away between the two species. And so why is this a problem for regression? In general, in regression, we have a variable y here that we write as alpha x, so a coefficient of proportionality times x, a variable that is observed, plus an intercept that we often write beta, plus epsilon. And we say that epsilon here is the noise. So the value of the variance of epsilon here gives us the amount of variation that is not captured by the model. We have a part here which is deterministic, which says that the value of y is a deterministic function of x here, plus some noise here, and the bigger is the variance of this noise, the less good of a fit is this particular function here compared to the actual values of y. And so one of the main difficulties with this approach is that we assume that the noise is independent in general. So it is really crucial for the linear regression and all kinds of regression that the variables here that are captured by this epsilon correspond to completely independent random variables. That is to ensure that there is no dependence between the records. But when we have a case of variables that we take from a biological tree, we know that this is violated. So we cannot really use the standard regression strategy because the amount of variation that we have between the records is very much dependent on each other. We see here in this example that A and D, the amount of noise between the two is actually very much dependent. Wherever is A, D is extremely close by. And the same can be said with this representation here. If the split between any two species is not long ago, then we know that one of these two species, in terms of the value of x that is measured here, has to be close to the other. Therefore, there is a strong dependency between the records, and we cannot use the standard linear regression approaches. In reality, this is not exactly true. You are free to use a standard linear regression package and then plug the value like this, and you are going to find a certain straight line here. And in a sense, this is a problem because that straight line here has been badly estimated. You will get an estimate for the values of alpha and beta, but because your assumptions are false, then these estimates are going to be very far from the actual measurements that you should make. So what can we do about it? One of the most straightforward approaches that we can take is to say that instead of assuming that the noise is independent, we're going to say that the noise is dependent. So we're going to allow the variations here to depend on each other, to say that if I have one point here and not a lot of time has passed, the other point is going to be kind of close by. They cannot be very far away from each other. And so to do this, we're going to take the approach of trying to directly model a certain amount of correlation that exists between the records, depending on how long they have been diverging. And so if we focus on this picture here, we can see that we can make a sort of distance from all the species, A, B, C, and D, that depends on the amount of time that has passed between the divergence of the species. So we can create a distance matrix here with the species A, B, C, and D, and we can fill the entries that correspond to the time that existed for them to diverge. So between A and A, the time is 0. And more generally, for every pair of species, B and B, C and C, and D and D, the distance is 0. It's exactly the same species here. So we can fill the diagonal very easily in that case. What about A and B? For A and B, this corresponds to the time that is here in red. And I would write this, for example, as T1 for time 1. For A and C, this is the time here which is in cyan. So these species have been diverging for a longer time. And so we would say that this here is T2. And I would fill the matrix with the value of T2 here. And likewise, I can fill the matrix here with all the values that I can take from the tree. But for this, I must make sure that I have the entirety of the tree here. If I know the topology of the tree between A, B, C, and D, and more importantly, if I know the amount of time that separates the species, then I can fill that matrix completely, and I would call that D for distance. So I'm able to compute a certain distance matrix that represents the amount of time that has passed between the divergence of each pair of species in the matrix. And then I could say that the correlation decreases over time. I could say, for instance, that it could be a function like exponential minus rho square times t. And rho would be a parameter that I would need to estimate, and the fact that I'm squaring it just guarantees that this is a positive parameter. So with a function like this, I would see that when t is equal to 0, the correlation is 1, which means that the information is 100% conserved from one species to the next, and as that time goes by, then the correlation would slowly or fast decrease to 0, so that given sufficient amount of time, I would have no influence from one species to the next because the divergence was so long in the past. This is it for the correlation. And for the covariance, I would multiply by a positive constant that I would call, for example, eta squared. And so that here would give me a particular function that gives the amount of variance as the time increases. Eta here plays the same role as the variance of epsilon here. If eta is high, it means that there is a large amount of noise, and if eta is small, it means that the amount of noise is much lower. That function here is just one possible way in which we could model the way that the correlation or the covariance decays over time. With this function, we have that at time equals 0, the covariance or the correlation is maximum, and as the time increases, the correlation or the covariance goes to 0. But there are other functional forms that we can use. For example, we could use eta squared exponential minus rho squared t squared. That's just another way that we could model that particular decay here. Any function where we have the fact that the correlation decreases to 0 as time goes by, and that is equal to 1 as the time is equal to 0, would actually work. So we have a large choice of the different functions that we can use for this decay here. And this is an important point. We have multiple options to model the decay of the correlation, and unfortunately this matters quite a lot. Some authors have initially proposed to use that particular formula here, which is based on the Brownian motion, a little bit idealized with this sketch over here, but over time people have said that this is probably not a very good model for biological evolution, and we should go away from that particular expression here and use other ones. This is one that has been proposed more recently as being more appropriate, but in general, one has to realize that this is probably a weak point of these strategies, in the sense that the choice of one or another form here is quite crucial, and yet we don't have a very good idea of which is the most appropriate for the problem at hand. So in this class we're going to use this one here, the one where we have only a linear decay over time inside the exponential. But this is by no means the only option, maybe not even the best. And so what do we do with this? Well, if we go back to the standard linear regression approach, we can see that in general, when we have a model like y is equal to alpha x plus beta plus epsilon, we're saying that y here is distributed as a normal variable with mean alpha x plus beta and with variance sigma squared. And sigma squared is the variance of the noise here, of epsilon. So we have the fact that epsilon is distributed as a Gaussian random variable with mean 0 and variance sigma squared. So we express the fact here that the noise in this particular equation is actually the noise of epsilon here. It's this variable that adds the noise. And this is formulated in the fact that now we have this term that represents the amount of noise in the model. Now when we cannot use epsilon anymore because this here is only working for uncorrelated variables, we can change this particular model to make it work in the case of correlated variables. And the change is actually fairly minimal. We can say that y here is a Gaussian distribution, but instead of a standard unidimensional distribution, we take it as a multi-dimensional Gaussian. And to symbolize this, I'm going to use capital letters here to say that this is a matrix multiplication of a and x plus another matrix here of intercept terms. So we have multiple terms, for example 4 in the examples that we gave, and here instead of the sigma that we have here, we have an entire correlation matrix, which is here, or a covariance matrix, and we're going to use this one here. So we would say that the covariance matrix is eta squared exponential minus rho squared times d. So here note that d is an entire matrix, here is the matrix which is there. So that means that all the terms that we have here are matrices. This here is actually a vector of 4 elements, and here this is a matrix of 4 by 4 that contains all the terms of covariance between all the species that are here. So there's a strict parallel between this first line here, which corresponds to the linear regression in the case of one single element, assuming that they are all independent, and a case of linear regression where we allow the variables to be correlated. That here is not just a simple Gaussian, this is a multivariate Gaussian or multivariate normal. So this is a distribution which is entirely parameterized by one vector of averages here and one matrix of covariances. So how does it work in practice, and what do we do when we actually want to fit a model like this? Well, this is pretty much exactly as usual. We had a model here with three parameters, that was alpha, beta, and sigma, or sigma squared. And now we have a model with alpha, but I'm going to write here A, because this is what I wrote in this particular expression. We also have another parameter which is a beta, but I'm going to write B as here. And we have two other parameters, eta squared and rho squared. So we went from a situation having three parameters to a situation with four parameters. And the parameters that we had before were A or alpha, B or beta, and here eta squared is really the equivalent of the sigma squared that is here. The new parameter that we have is rho squared, which represents the amount of decay here of correlation over time. This is something new that I need to incorporate in the model, but overall, look at it, we just have one extra parameter. So we can deal with that situation if we accept to have one extra parameter. And so what do I do with that in practice? Well, as usual, it's always the same story. We need to get the priors over these parameters. So I need to find good priors for alpha, beta, eta, and rho. And after this, I just use the standard approaches to get to the posterior distributions of all these parameters. So one task is to find good priors for alpha, beta, eta, and rho. And in general, I had good priors for alpha, for beta, and for eta because they correspond to the usual situation that we have here. We must make sure that what we choose for eta and then by extension for rho are priors over values that must remain positive. So we have to choose distributions like the exponential, the half normal, or the half Cauchy that have only positive values. And otherwise, there's not so much difficulty. The data consists of x, y, and d. And so I can use the standard techniques that we have developed so far so that we can process this data here and then sample the parameters there to produce the output y. Here it's really important to say that x and y are data as usual, so they correspond, for instance, to genome size or GC content, and d is also data. This knowledge must be completely available to me and there cannot be any mistakes with that. Otherwise, I'm going to misestimate the correlations or the covariances, and I'm going to make mistakes in the analyses. So it is really critical that D is fully available at the time I'm starting this kind of analyses here. So we can use the standard approaches where we use either NUTS or SVI. Both of them are going to work in these cases. And we are going to be able to make a model that expresses these parameters here and as a function of these particular observations of the data x, y, and d. And so the most important thing to understand is that this is pretty much exactly the same as a standard regression here, except that we have modified the term there. Instead of picking just one value here, which is sigma squared, that would be a parameter that we sample if we use a standard linear regression strategy. Here we sample two parameters, eta squared and rho squared, and we compute with a very simple expression here the value of the covariance matrix in that particular expression. And that, in principle, should allow me to proceed exactly as before with the methods that we have seen so far. So strategically, here we're doing one important step, which is to incorporate the unknown into the model. We have a problem that, for instance, we assume that there is independence between X and Y, between the records, but this is not the case. So what do? We then start to incorporate the fact that this is not the case directly through the knowledge of D. So we add one more parameter here, which is rho squared, that specifically captures the dependence between X and Y into the model. It's really important to understand that we really try to model the noise here. We try to model the correlation between X and Y, and by doing so, we're going to get a much more accurate result, because we're not going to make a mistake about the value of the noise here, and therefore this absence of mistake is going to give me much better estimates for everything else. Otherwise, if we're assuming a model here which is too simple, but that in practice is violated, which means we assume independence between the observations, but this is not the case, then we make an error here on that term, and this error is dispatched on all the rest of the model, and this could have totally unpredictable effects. So here once again, the main idea is when something is violated, a very standard strategy is to incorporate the knowledge that we have of this violation into the model. In that particular case, it's the fact that the records are not independent, and we specifically introduce here the dependency between the records. And that of course has to be correct. If we make a mistake here, then instead of solving the issue, we're actually making it worse because we put wrong information here. Another comment that I'd like to reiterate is the fact that here the choice of the function we have for the decay of the correlation seems to be quite important. Whether we use that variant or another one will actually have some effect on the estimates of alpha and beta here, for instance. So it's quite crucial to understand what do we model when we use one or the other, and also to experiment a little bit with different versions, just to see how sensitive the conclusions are to the assumptions that we make. And that's what I wanted to share with you. Thank you for listening.